Hello and welcome everybody to my channel UoH Bioentrance, and today we will be discussing University of Hyderabad PhD entrance examination biochemistry question paper of the year 2024. If you are new to my channel, please do like, share and subscribe. I have also uploaded previous year's biochemistry PhD entrance examination question paper of University of Hyderabad. You can get the same in my channel. Coming to the first question. Triploids are not fertile because gametes are diploid for all chromosomes are formed that cannot fertilize. Gametes are haploid for all chromosomes are formed that cannot fertilize. Gametes are diploid for some chromosome and haploid for others. So they cannot fertilize. So answer is option C. Annealing temperature in PCR needs to be. So the annealing temperature of a reaction, that is, it is usually lower than the melting temperature to ensure primer hybridization to the template. Now if the annealing temperature is too high, the primer will not anneal to the template and amplification will not proceed. If the annealing temperature is too low, non-specific binding of the primer to the template can occur, and or it can even result in the formation of primer dimers. So annealing temperature, it has to be slightly lower than the melting temperature of the primers. So answer is option A. Now next one. The slowest migrating band on the gel for plasmid preparation represent. So if you see the photograph that I have given here, you can see that the plasmid various conformations, that is nicked, linear, supercoiled, circular and single stranded. So what is supercoiled plasmid? That is the conformation that is found in vivo and it migrates faster in an agarose gel. What is nicked? Nicked happens during replication. So cellular topoisomerases, they nick one strand of the DNA helix to relax the superhelical tension, and thereby it favors the binding of the DNA polymerases. So this is the slowest migrating form in an agarose gel. Then linear, it occurs when the DNA helix is cut in both the strands at the same place, and it is found between the nicked and the supercoiled forms. Then circular single stranded plasmid. This happens when you have some when you are isolating plasmid DNA by alkaline lysis step and you incubate the plasmid DNA for too long in the alkaline lysis buffer, then the DNA can become permanently denatured and give rise to single stranded closed circles that will migrate ahead of all the other forms of the plasmid. That is called as circular single stranded. So in the question they have asked the slowest migrating band for plasmid preparation is the nicked form of the plasmid. So answer is option C. Then PAM, protospacer adjacent motif concerning CRISPR is present on the. So PAM is protospacer adjacent motif. It is found adjacent to the NGG. It is found adjacent to the protospacer. NGG is the PAM, that is protospacer adjacent motif, and it is found adjacent to the protospacer. This is PAM, this is protospacer, and it is a sequence of about 2 to 6 nucleotides long, and it is found on the viral genome and not a component of the bacterial CRISPR locus. So the answer here is option C, that is target genome. Then in RT-PCR, what is the relation between CT values obtained and amount of RNA present in the sample? So what is the CT value? So CT value, it represents the cycle number at which the sample reaction, this is the cycle number at which the sample reaction crosses the threshold, indicating detection of the target nucleic acid. So these are the CT value. When it crosses the threshold, what is the cycle number that represents the CT value? Now if lower CT values, that is at lesser cycle number, if you see lower, it indicates higher target sequence amounts, and while higher CT values, it indicates lower amounts of the target. So the answer here is option C, inversely proportional. That is, CT value obtained and amount of RNA present in the sample is inversely proportional. Answer is option C. Then next one. You plan to clone your favorite gene YFG in a bacterial expression vector PE at the BAM-H1 site. The size of YFG and PE are 0.5 kb and 3 kb respectively. Your advisor suggested you to keep 1:3 vector insert ratio in the ligation mixture. Which of the following will give you the desired stoichiometry? So, they have given vector amount in nanograms, that is 100 nanogram, that remain the same. Then they have given size of YFG and PE plasmid and favorite gene of interest sizes they have given. Then 3:1 vector insert ratio. Now they have asked which of the following will give you the desired stoichiometry. So the amount of insert DNA required, that is nanogram of insert, that is what you have to find out. So nanogram of vector into kilobase per size of insert by kilobase per size of vector into molar amount of insert by molar amount of vector. Now substitute the values. Nanogram of vector, they have given it is 100 nanogram. In all the option it is 100 nanograms. Kilobase size of insert, it is 0.5 kb. Kilobase size of vector is 3 kb. And molar amount of insert by molar amount of vector. So here they have given 1:3 as vector insert ratio. So 3/1. So it will give you 50 nanogram of insert. So answer here is vector 100 nanogram and insert 50 nanogram. Answer is option A. Then next one. A sequence of amino acids in a certain protein is found to be serine, glycine, proline, glycine. The sequence is most probably part of. So proline and glycine, they are found in the beta turns. So the sequence here, it is probably part of the turn. So answer is option B. Then next one. The outer membrane of mitochondria is permeable to ions and solutes with molecular weight less than 10 kDa is due to the presence of. So outer mitochondrial membrane, it is freely permeable to small molecule because it contains the protein called as porins. Sorry. These are channels that allow free diffusion of the molecules. And what they have asked here, the outer membrane is permeable to ion and solutes with molecular weight less than 10 kDa is due to the presence of porins. Answer is option B. Then in urea cycle, the enzymes are compartmentalized in mitochondria and cytosol because no specific reason to prevent reductive amination of alpha ketoglutarate in mitochondria. That is the correct answer. Answer is option. Then next one. What is the fraction of deprotonated histidine at pH 7? The pKa of histidine is 7.4. So you know the Henderson-Hasselbalch equation. pH is equal to pKa plus log concentration of A minus by concentration of HA. That is the concentration of deprotonated form divided by protonated form. Now substitute the values. You get 7 minus 7.4 is equal to log concentration of A minus by HA, that is minus 0.4 is equal to log concentration of A by HA. Antilog of minus 0.4 is 0.4, or A is equal to 0.4 HA. Now A plus HA concentration is equal to 1 molar. Substitute the values and you will get it as 71%. But they have asked for the deprotonated histidine. So you have to find the value for A minus. Now you have got the values for HA only. So for A minus, you substitute the value of HA and you will get it as 29% or 0.28 as in option A. So the answer here is option A. Then next one. One reason why vaccines fail to work in very young infants is the presence of. So it is due to the presence of maternal antibodies. Because maternal antibodies inhibit the development of the infant humoral response to both live and killed vaccines. Then next one. Which one of the following hydrogen bonds best represent the type of hydrogen bond that keep the helix and the sheet in a protein from falling apart? So hydrogen bonding between carbonyl group and the amino group of amino acids. So it is the NHOC bond as in option D. Then next one. Match the columns correctly. Glucose 6-phosphate dehydrogenase, it is okay, they have given it in the reverse way. Okay. Phenylketonuria. Phenylketonuria is for phenylalanine hydroxylase. Phenylketonuria, phenylalanine hydrolyse. So 1B. Then irreversible reaction, it is pyruvate kinase. 2C. Then HMP shunt pathway, it is hemolytic anemia. Because hemolytic anemia, it is due to the deficiency of HMP shunt enzyme, that is glucose 6-phosphate dehydrogenase. So 3. HMP shunt pathway is hemolytic anemia. And 4. NADPH, it is phenylalanine, glucose 6-phosphate dehydrogenase. So 16 answer is option A. 1B, 2C, 3D and 4A. Then next one. A protein was purified from anaerobic bacteria and analyzed by polyacrylamide gel electrophoresis containing SDS. Following protein staining, a single band was observed. The same protein was analyzed on a second electrophoresis under native conditions, that is non-denaturing or without SDS. This gel shows 2 bands after staining. Assuming no errors was committed during these experiments, what is the best possible explanation about the protein? So here SDS showed only single band and native page showed 2 bands, showing that the 2 proteins of identical molecular masses. These 2 proteins will also differ in their pi. SDS does not disrupt the disulfide bonds. So 2 identical polypeptides with disulfide bond can be omitted. So it is 2 proteins with identical molecular mass having different pi. So the answer here is option B. Then next one. Which group of peptides are produced after cleavage of the following peptide with trypsin? Now trypsin cleaves the peptides on the C-terminal side of lysine and arginine amino acid residues. So here if you see the sequence of protein that they have, of the polypeptide that they have given, you can see arginine occurs here and lysine occurs here. So on the C-terminal side of arginine you will get a cut, and similarly for the lysine also you will get a cut with trypsin. So what are the fragments you get? That is met-alanine, tyrosine, met-phenylalanine, arginine will be one. Glycine, aspartic acid, lysine will be the second, and glutamic acid and TRP will be the third. So you will get three fragments, and this fragment option you will find in option A. Met-alanine, tyrosine, met-phenylalanine, arginine, that is one. Glycine, aspartic acid, lysine is second, and glutamic acid and TRP is the third. So the answer here is option A. Now, which of the following ionic species of glutamate would be prevalent at pH 9? So this is the structure of glutamic acid. It has got two COH bonds, COH carboxylic groups. One is the alpha carboxylic group and the other is the side chain carboxylic group, and there is one NH2 group also. So it has 3 pKa values. For the alpha carboxylic it is 2.1, for the side chain carboxylic it is 4.1, and for NH2 it is 9.5. Now at pH 9 we will see. At pH 9, pK value is less than pH 9. So this will be deprotonated and it will gain a net negative charge. So when coming to the side chain carboxylic group, pK will be 4.1. So it is also less than pH 9. So it will also get deprotonated and it will gain a net negative charge. Now amino group, for the amino group pKa is 9.5. 9.5 pKa is greater than pH, so it will be protonated and this will get a net negative positive charge. So if you see the options, you can see that option C is the right one. Here there is a minus for carboxylic group side chain, and alpha carboxylic group also minus, and then amino group is positive. So the answer here is option C. Now next one. Since you have performed non-direction cloning of your favorite gene, which is 0.5 kb at the BAMH1 site of the expression vector PE, you want to check the orientation of the insert. To that end, you wanted to use restriction enzymes whose sites are present once in the insert and once in the vector at the multiple cloning sequence as shown in the figure. The EcoR1 site is present at 100 base pair downstream of the start codon, while the SAL1 site is present 250 base pair downstream of the start codon of the gene. Which of the following statement is correct? Now what they have told is you have a gene called as your favorite gene or YFG. It is 0.5 kb in size. And you have cloned this gene at the BAMH1 site of an expression vector PE, which is of 3 kb. But you don't know whether the orientation of your gene of interest is in the right way that you want to the gene getting transcribed and translated to proteins. But if it is the orientation of the gene is in the reverse direction, that is not promoter, then your start sequence, then the stop codons. If it is not in that order, then you will not get a protein product. Now what you do is to check the insert, you will digest it with different restriction enzymes. You will digest it with different restriction enzymes. So first, okay, I have done it in the next slide. Okay, now if your sequence that they have told from the start sequence, EcoR1 site for your gene of interest is at 100 base pairs. and the SAL1 site is at 250 base pair from the start site. Now this is your start. So eco R1 will be 100 base pair and SAL1 site will be 250 base pair from the start region. So this is the eco R1 of the vector. This is the eco R1 inside your gene of interest. This is SAL1 in your gene of interest and this is SAL1 of the vector. Now the whole sequence of this particular genome is 500 base pair. Now what they have told is, if the insert is in correct orientation, digestion with eco R1 will give you 3 fragments. No, you will get only 2 fragments, because after digestion with eco R1, this sequence will be removed off. This will be 100 base pair. They have already mentioned from the start of a sequence to your there is a 100, after 100 base pair from the start region there is an eco R1 site. So this will be 100 base pair and 100 base pair will be cut off. The remaining will be 500 minus 100, that is 400. That is this portion. This is 400 portion plus this 3 kb and plasmid DNA. So that will be 3000 base pair. So 400 plus 3000 will give you 3400 base pair. Now if it is in the reverse orientation, that is the start sequence will come over here, this side. So the start, then you have got the eco R1 site which is at 100 base pair from the start, and then you have got the SAL1 site which is 250 base pair from the start, and the whole sequence gene of interest is 500 base pair. So once you use eco R1 for digestion, it will cut over here as well as here. So this whole sequence will be 400 base pair. And what will be the remaining? 100 base pair plus 3000 base pair of the plasmid. So 400 base pair plus 3100 base pair is what you will get when you digest with eco R1 if the orientation of your gene of interest is in the wrong way. So the answer here is, if the insert is in correct orientation, digestion with eco R1 will give you 2 fragments, 400 base pair, 3. No, that is not the correct one. So if the insert is in the wrong orientation, digestion with eco R1 will give you 2 fragments, that is 400 base pair and 3100 base pair. So the answer here is option C. The next one. The binding affinity of a protein for its ligand at pH 7 and room temperature is 2 into 10 raised to 5 per molar. At what ligand concentration will be 80% of the protein bound? So you know theta is equal to concentration of L by concentration of L plus Kd, where L is concentration of the ligand. Now they have given the value of theta, that is 0.8%, that is 80%, so it is 0.8. So is equal to L by L plus Kda. Now you substitute the value, multiply both sides with L plus Kd, then you will get 0.8 Kd plus 0.8 L is equal to concentration of L. 0.8 Kd is equal to L minus 0.8 L, or 0.8 Kd is equal to 0.2 concentration of L. Now Kd, they have given the binding affinity, that is Ka is equal to 2 into 10 raised to 5. So Kd is equal to 1 by Ka, that is equal to 1 by 2 into 10 raised to 5. So 0.8 into 1 by 2 into 10 raised to 5 is equal to 0.2 concentration of L, that is 0.4 by 10 raised to 5 is equal to 0.2 concentration of L. 0.4 by 10 raised to 5 into 0.2 is equal to concentration of L. 2 by 10 raised to 5 is equal to concentration of L, that is equal to 2 into 10 raised to minus 5, or 20 micromolar. So the answer here is option A, that is 20 micromolar. Then next one. During lagging strand synthesis, to join together two precursor fragments, several enzymatic activities happen sequentially. Lagging strand synthesis, Okazaki fragments are formed, and these Okazaki fragments, they have to be joined together in the end. So for the following, pick the relevant activities and arrange them in correct order. Note that the two Okazaki fragments are already synthesized. So what are the various enzymatic activities that will be taking place? 5' 3' polymerase activity of DNA pol 3, that is not required there. 5' 3' helicase activity of DNA B is also not required. 5' 3' polymerase activity of DNA polymerase 1 is required. 3' 5' exonuclease activity of DNA polymerase 3 is not required. Ligase activity is required. 5' 3' exonuclease activity of DNA polymerase 1 is also required. So 3, 6 and 5. So the answer is option B. Then next one. The frequency of two alleles in a gene pool of a population that is in Hardy-Weinberg equilibrium is 0.19 and 0.81. The percentage heterozygous and homozygous recessive individuals in this population is. So frequency of allele A is 0.19, frequency of allele small a is 0.81. So according to Hardy-Weinberg equilibrium, P square plus 2PQ plus Q square is equal to 1. P square is frequency of homozygous dominant genotype. 2PQ is frequency of heterozygous genotype. Q square is frequency of homozygous recessive genotype. So percentage of heterozygous individuals, that is 2PQ is equal to 2 into 0.19 into 0.81 is equal to 0.3078 or 31%. That is percentage of heterozygous individuals. And percentage of homozygous recessive individuals, that I have not done here. I forgot to add that one, but I will tell you how to do it. So percentage of homozygous recessive individuals is Q square. So 0.81 into 0.81, that will give you around 0.6561. So that will be 66%. So the answer here is option B, that is 31 and 66 respectively. Then the P/O ratio, P is to O ratio, is a measure of how many moles of ATP are formed from ADP per gram. That is the number of phosphates that are fixed into ATP for every 2 electrons that pass from a substrate to reduce each oxygen atom of molecular oxygen. So atom of oxygen for a given substrate. Answer is option A. Which of the following is not an electron carrier of the electron transport chain? It is NADP. Answer is option C. Whereas NAD, coenzyme Q, FAD, they are all electron carriers in electron transport chain, but not NADP. Then the addition of valinomycin to the active respiring mitochondria in the presence of potassium dissipate. So valinomycin, it binds to K positive ions on the surface of the mitochondria, and it will form what is called as the K plus valinomycin complex. This complex is highly soluble, and it will highly soluble in the and it will cross the membrane, mitochondrial membrane, and once inside, K positive is released and the valinomycin is free to return and it can bind another K positive ion. This will result in the disruption of the normal potassium gradient, and this action, it increases the permeability of the membrane to K positive, and it will lead to dissipation of the electric potential across the membrane. This reduction in electric potential, it affects the proton gradient, which in turn impairs the ATP synthase activity, leading to decrease in ATP production. So the addition of valinomycin to active respiring mitochondria in the in the presence of potassium dissipates electric potential followed by proton gradient is the answer. Answer is option D. Then next one, inappropriate antidiuretic hormone secretion can be associated with which of the following events? Diabetes mellitus insipidus, hyponatremia, water intoxication. So what is hypodiabetes mellitus? Everybody know. Insipidus also it's caused due to lack of ADH or the kidney's inability to respond to ADH. It causes diabetes insipidus. Hyponatremia. It is low level of sodium in your blood. It happens when your body make excess amounts of ADH, and this causes your body to retain too much water. So diabetes insipidus and hyponatremia is the answer for inappropriate antidiuretic hormone secretion. It can be associated with diabetes insipidus and hyponatremia. So the answer is option 2, that is B, 2 and 3. Then next one. Non-enzymatic reaction between the carbonyl group of glucose with the amino group of lysine R chain is known as. So it is called as Maillard reaction. Answer is option C. Then next one. Which of the following derivative of the bile contributes to the color of the feces? So it is stercobilin. That is, answer is option D, stercobilin. Then next one, hormone that antagonizes the insulin action and is also considered diabetogenic. So it is the growth hormone. It promotes diabetogenic action by antagonizing insulin action in muscle and adipose tissue. So it is growth hormone. Answer is option D. Then during nerve conduction, action potential and Na plus K plus ATPase pump work in coordination. Identify the correct statement about both events. Both events pump Na positive in and K positive out of the cell. That is not correct. During an action potential, Na positive ions move in and K positive ions move out, and the Na plus K plus pump exports Na positive ions out and K positive in. That is correct. Answer is option B. Gestational diabetes could be due to. Now this question they have asked for PhD Biochemistry entrance examination conducted in the year 2023. So it is due to high placental lactogen secretion. Answer is option C. Then palmitoleic acid is a monounsaturated fatty acid. Answer is option B. Then cholesterol, it is an animal sterol. It is not found in plants and microbes. So answer is option B. A futile cycle or substrate cycle. So a futile cycle occurs when two metabolic pathways run simultaneously in opposite direction and have no overall effect other than to dissipate energy in the form of heat. That is called as futile cycle or substrate cycle. Now even if they dissipate energy in the form of heat, they have roles in regulating various metabolic processes and also for maintaining homeostasis. So a futile cycle or substrate cycle may have a role in regulating metabolic process. That is correct. Dissipate energy in the form of heat. That's also correct. Maintains energy homeostasis. That is also correct. And D is all of the above. So all of the above is the answer. Then complete hydrolysis of glycerophospholipid yield glycerol, palmitic acid, oleic acid and phosphoric acid in the molar ratio 1:1:1:1. The lipid that was hydrolyzed was. So it is phosphatidic acid. A similar question was asked in the year 2023 also. Answer is option A. Then next one. The direct product of beta oxidation of a fully saturated straight chain fatty acid of 17 carbons is. So odd chain fatty acids. Now 17 carbon, it is an odd chain fatty acid. They also undergo beta oxidation in the same manner as even chain fatty acids. But once a 5 carbon chain remains, the final spiral of beta oxidation will yield one molecule of acetyl CoA and one molecule of propionyl CoA. So here if you see, this is the spiral of the beta oxidation. So first 2 carbon atoms are oxidized, then comes 4, then 6, then 8, then 10, then 12. So 5 are remaining after 12. They will form acetyl CoA and propionyl CoA. Then acetyl CoA, then after each turn of the cycle, acetyl CoA will be released. So how many acetyl CoA are formed? 1, 2, 3, 4, 5, 6, 7 acetyl CoA and 1 propionyl CoA. So the answer here is option A. Now they have asked a similar question in 2023 also. Then when a protein is denatured by heating, the absorption as measured in UV light will increase or decrease depending upon the wavelength. So answer is option C. Major interaction for protein structure stabilization, it is your hydrogen bond interactions. So answer is option C. If you were given to purify protein A from tissue by employing different techniques, how will you check the purity of protein A? That is by SDS page only. Affinity assay of protein is all of the protein you are assaying it by Bradford assay, by Urette assay, like that. SDS page you will get different bands at different showing the different molecular weight of the protein. By affinity chromatography you are purifying. By spectrophotometer it is the same as somewhat same as assay for protein. So the answer here is by SDS page. Answer is option B. Polyuria can occur in diabetes mellitus diarrhea. The answer is option B. Serum cholesterol is decreased in. So it is decreased in thyrotoxicosis. Answer is option B. Phenomenon that some cells evoke a specific developmental response in other cell is. So it is embryonic induction. All these are direct questions. Then blastoderm split occurs in gastrulation. Answer is option D. BLAST-N is. So BLAST-N can be used to compare a nucleotide sequence with a nucleotide database. So it is used for pairwise sequence alignment tool. Answer is option C. Then KEGG is primarily it is a pathway database. Answer is option B. Then what is the expected ratio of genotype in an F2 progeny of a monohybrid cross when one of the alleles is dominant? So expected ratio of genotype in F2 progeny of a monohybrid cross. So after the monohybrid cross, the F2 progeny will be capital T small t into capital T small t. These are your capital T small t are your F1 progenies. So once you cross capital T small t with capital T small t, you get the F2 progeny. So F2 progeny, the monohybrid expected ratio of genotype will be 1:2:1. So the answer here is option B. Then next one. Which one of the following statement is incorrect? Alleles are alternate forms of the same gene. That's correct. During meiosis, gametes inherit one allele of a gene. That's also correct. In a population, when individuals with the same combination of alleles show different degree of phenotype, it is a case of incomplete penetrance. That is incorrect statement. In a population, there are multiple alleles for the same gene. That is ABO blood grouping and all comes under that. So D is also correct. So what is incorrect is option C. Then ribosome assembly take place. So in the ribosome assembly take place in the nucleus. Okay. Actually, it is the nucleolus. Since nucleolus is present in the nucleus, nucleus can also be the right answer. Then next one. Glycosylation of protein happens in. So there are two types of glycosylation, N-linked and O-linked. N-linked take place in endoplasmic reticulum and O-linked take place in the Golgi. So the answer here is option B, that is endoplasmic reticulum and Golgi. Then which of the following statements is correct? Pair of they have asked phosphofructokinase 2 is a tandem enzyme, citrate synthase is a tandem enzyme, phosphofructokinase 1 is a tandem enzyme, hexokinase is a tandem enzyme. So which of these is a tandem enzyme? That is the question. So what are tandem enzymes? So they are pair of opposing enzyme activities that are resident in the same polypeptide chain are known as tandem enzymes. Now if you take the case of phosphofructokinase 2, it is a tandem enzyme. It has two domains that act as independently functioning enzyme. One of them serve as kinase domain. That is, it is having phosphofructokinase 2 activity. That is fructose 6-phosphate plus ATP getting converted to fructose 2,6-biphosphate plus ADP. And the other has got phosphatase activity. That is fructose 2,6-biphosphatase activity. So it will convert fructose 2,6-biphosphate to fructose 6-phosphate. So they are pair of opposing enzymic activities that are resident in the same polypeptide chain and they are known as tandem enzymes. So phosphofructose kinase 2 is a tandem enzyme. Answer is option A. Then, which of the following two enzymes are glyoxylate cycle specific enzymes? So they are isocitrate lyase and malate synthase. Answer is option B. Then next one. The core oligosaccharide added to asparagine residues in N-glycan synthesis contain. So it contains 2N-acetyl glucosamine, 9 mannose and 3 glucose residues. Answer is option C. Then in the urea cycle, which of the following is correct for the formation of one urea molecule? It is for one urea molecule in urea cycle you need 3 ATP molecules. Answer is option B. Then glutamine synthetase is regulated reversibly by adenylation and deadenylation. Answer is option A. Which of the following is a glycosylated form of hemoglobin used for assessing long-term blood glucose control? So it is HbA1c we call it. Normally it is hemoglobin A1c. Answer is option B. Which cardiac marker is considered specific for myocardial infarction? So it is troponin. Answer is option C. So that's it. Thank you for watching my channel. Please do like, share and subscribe. I have uploaded previous year's PhD entrance exam question papers also. You can go through my channel playlist for the same. Even for biochemistry as well as biotechnology I have uploaded. So thank you. Please like, share and subscribe.